Good afternoon, everyone. Today, I'll be discussing about exercise 9 that were performed on February 28, 2020, which is entitled Extraction of Lipids from Brain Tissues. The objectives of this experiment is to first define saponifiable and non-saponifiable lipids, including their structures, properties, as well as their functions in the human body. Second, we want to relate these definitions to the procedures of isolating saponifiable and non-saponifiable lipids from brain tissues. We also want to analyze the reagents used in different parts of this procedure. And lastly, we want to evaluate the factors that could have affected the yield of lipids from brain tissues. So first, we want to define what are lipids. Lipids are composed of fatty acid chains with formula CH3, CH2N, COOH, where N ranges from 10 to 28. It is classified into a saponifiable and non-saponifiable lipids based on their reactivity with strong bases, which is saponification, and I will discuss that later. To answer post-laboratory question number 8, what are neutral fats? Neutral fats are fats that don't have acidic or basic groups. They are also amphiphilic, which means that they have both polar and nonpolar groups, and are hydrophobic in nature. They are soluble in nonpolar solvents such as esters and hexane. So, the first lipid that I will discuss is saponifiable lipids. Saponifiable lipids are lipids that have an alcoholic functional group connected by an ester linkage. When it reacts to strong bases such as sodium hydroxide, the ester link undergoes saponification along with the fatty acid chain, leaving only the alcoholic functional group, which, with the example I showed to you, the triglyceride, that was discussed last experiment, the alcoholic group is glycerol. So when the triglyceride goes under saponification, by the end of it, you obtain a glycerol plus a crude soap. And under saponifiable lipids, there are three major groups. One is triglyceride, and the second one is glycerophosphatides. Glycerophosphatides are more commonly known as phospholipids. It has an alcoholic functional group which is also glycerol, and it has two fatty acid chains connected to carbon 1 and 2. And the third carbon, a phosphate group, is connected, which is why the name of the lipid group is glycerophosphatide or phospholipid. This is the general formula of glycerophosphatides, which answers the post-laboratory question number 5. So, phospholipids make up the phospholipid bilayer, which is the main component of the cell membrane. So, remember ang phospholipid kay nasa polar head o nonpolar head. Non-polar tail. The tail is non-polar because it is composed of two fatty acid chain. However, the head is polar because it is a phosphate group. So that is how they make up the permeable membrane, which is the phospholipid bilayer. Its main function is to regulate what goes in and out of the cell, allowing the cell to proceed with their activities separate from its surrounding environment. So, the last one under the saponifiable lipids is the sphingolipids. The sphingolipids, unlike phospholipids and triglycerides, has a sphingosine as its backbone instead of a glycerol. A sphingosine is a 3-carbon molecule which has an alkyl group in its first carbon, an amine group in its second carbon, and a hydroxyl group in its third carbon. So, the general formula of a sphingolipid is a sphingosine with a fatty acid chain in its second carbon attached through an amide group and a group in the third carbon attached by an ester linkage. So depending on what group is attached in the third carbon, determines their uh, group structure under sphingolipids. So there are two structures, the sphingosine phosphatides and sphingosine glycosides. The sphingosine phosphatides are called that because they have a phosphate group in the third carbon. They are most commonly known as sphingomyelins. Its general formula is a sphingosine connected with a fatty acid chain on carbon 2 attached through an amide bridge, and on carbon 3, a choline is attached by a phosphate group. To answer post-laboratory question number 7, what can be said about the structure of sphingosine phosphatide? As you can see, there are both positive charge and negative charges and a negative charge. So that means the lipid is a deuterion. Being a deuterion, they are really good in hydrogen bonding, which allows them to form lateral domains. So, sphingomyelins are building blocks of the membrane, most commonly found in myelin sheaths. Thus, the name sphingomyelins, ang myelin sheaths kay sila ang mag-protect sa axon sa kanang nerve. Unya, maong abundant ang sphingomyelin sa myelin sheath kay sila ang ga-form anang lateral domain as shown in the unwrapped myelin. So, sphingosine is significant in protein sorting, regulation of endocytosis, and mag-act sila as receptors of various toxins. And because of the choline present, sphingomyelins also help increase the integrity of cell membrane by participating actively in cell signaling, modulation of gene expression, lipid metabolism and transport, and early brain development. The other structure is sphingosine glycoside. Diba, ang sphingosine phosphatide kay phosphate ang naka-attach sa iyahang third carbon, which is why ginatawag siya sphingosine phosphatide. Ang sphingosine glycoside kay ang attach sa iyang third carbon is a glycoside or a carbohydrate, usually a monosaccharide. Sphingosine glycosides are more commonly known as glycolipids. To answer post-laboratory question number 7, what can be said about the structure of a glycolipids? Ang structure niya is amphiphilic, na both polar and non-polar group. Sphingosine glycosides are also abundant in myelin sheaths and are called cerebrosides kay abundant sila kaayo sa white matter sa brain. They help provide energy and help in cell recognition because sa carbohydrate na present sa ilaha. So on the other hand, so that was the end of the saponifiable lipids. On the other hand, the non-saponifiable lipids are lipids that don't contain ester groups, so they cannot react with strong bases and they cannot be saponified. This includes cholesterol and other fat-soluble fatty acids. So, cholesterol. Cholesterol is a sterol because it is composed of a steroid group and an alcohol group. To answer post-laboratory question number 2, the general structure of cholesterol is an alcohol group, a steroid group, and a fatty acid chain. So, what is the function of cholesterol in the human body? Cholesterol helps in cell membrane development, hormone synthesis, cell transport, and nerve conduction, which is why they are also abundant in myelin sheaths. So, why use brain tissues? Because after defining all three molecules, all three lipids that we are going to extract in this experiment, as you have noticed, they are present in myelin sheaths, and diba, brain relies on networks of nerves which are coated with myelin sheath, and since all lipids that we want to extract are present in myelin sheath, we are sure that there can be a lot extracted from the brain. So that is why the brain is a good source for lipid extraction. Now, we will go to the procedures of the isolation of cholesterol. So, what we did first is we triturated 20 grams of the pig's brain with 20 ml of acetone. It was then transferred to an Erlenmeyer flask and was added with 50 milliliters of acetone and was allowed to stand overnight in the refrigerator. The purpose of which is to successfully recover lipids and eliminate non-lipids by the acetone reacting with non-polar lipids through its polar group and also reacting to lipids with its non-polar group. It was then filtered and was washed with 20 ml acetone. The residue was set aside for the preparation of phospholipids and sphingolipids. So, by this procedure, how was cholesterol isolated? To answer post-laboratory question 1, only cholesterol goes down with acetone because saponifiable lipids can't be soluble in the acetone polar group, unlike cholesterol, which has a free alcohol group. So after filtering, the filtrate was concentrated to 10 milliliters through evaporation over a steam bath. It was then cooled in an ice bath. After cooling, cholesterol crystallized and was collected again by filtration. The filtrate was discarded and then the residue was used as it was the crystallized cholesterol. It was dissolved in minimal volume of hot ethanol filtering while the solution is hot and was then allowed to cool in a pre-weighed evaporating dish as a receiving vessel. So why did we use a hot ethanol? With the high temperature, ethanol is more efficient in extracting alcohol. Ethanol is a highly polar solvent and dissolves cholesterol because cholesterol is amphiphilic. It has both polar and non-polar groups. However, it is more leaned into polar group because of the alcohol of the free alcohol group. So after the isolation of cholesterol follows the isolation of phospholipids. The residue from before was used in this procedure. So the residue was extracted with 30 ml hexane or with 30 ml hexane. To answer post-laboratory question number 3, why can phospholipids be extracted by hexane? It is because hexane is highly nonpolar and phospholipids are amphiphilic, but favors nonpolar because it has two fatty acid chains unlike the others. So hexane will be effective in binding with the phospholipids. It was allowed to stand for 30 minutes with occasional stirring and was separated through filtration. The residue was used for the isolation of sphingolipids. So, it was concentrated over a steam bath. 30 milliliters of acetone was poured into the extract and was stirred. After that, the precipitate was then filtered off and it was dissolved in 10 milliliters of 1:3 methanol chloroform mixture. To answer post-laboratory question number 4, why was this solvent used? Is because no pure solvent is perfect for the extraction of lipids. So, as a solution, scientists mix chloroform and methanol because chloroform is highly polar and methanol is highly efficient with chloroform. So lastly is the isolation of the sphingolipids. So the residue from the last filtration from the isolation of phospholipids was extracted with 50 milliliters of hot ethanol. To answer post-laboratory question number 6, the head of sphingosine is highly polar and bonds with ethanol, eliminating unwanted residues. And ethanol is more efficient with high temperature. That's why it was used to extract sphingolipids. The mixture was boiled for 10 minutes over a water bath and was filtered and then the residue was discarded. The filtrate was cooled, and then the precipitate was collected by filtration. In a pre-weighed evaporating dish, the precipitate was dissolved by 10 milliliters of 1:3 methanol is to chloroform solution. With the same principle, this solution was used because it is close to perfect in extracting sphingolipids, as it was mixed, merging the properties of both methanol and chloroform. It was then recrystallized by evaporating the solvent, and then it was weighed. So this was the yield obtained by the different groups. Group 1 is Bea, Hans, Marty, and this was Group 2 is Krisa, Micah, and Ira's group, and then Group 3 is me, Brittany, Jericho, and Z. As you can see, we really obtained small amount of the lipids. The percent was obtained by dividing the obtained grams by 20 grams, which was the amount of sample used in this procedure. So it could be possible. The possible factors that could have affected this yield is, first, we didn't know how the tissue was gotten out of the body of the pig. So it might be possible that upon getting the brain tissues, the tissue experienced trauma, and by doing that, the lipids could have undergone lipolysis, which is where the fatty acid breaks from its backbones. Also, it might be possible that the active enzymes in the brain attack the lipids, as it wasn't refrigerated right after it was taken. We took the brain tissues around 1 o'clock in the afternoon, and most probably it was taken out of the pig's body in the early morning of that day around 5 a.m. And aside from that, from the market to the laboratory, it took 1.5 hours to get there. So the brain tissue was really exposed to not cold temperature, which is the supposed ideal condition for the tissue to not lose its lipids, or the lipids could have undergone lipolysis. And then lastly, we didn't know if the brain tissue was washed with water. If it was washed with water, then it might have affected the yield too. So with this, taking consideration of these factors, I think that it is within reason that we get as small amount of lipids as this. Also, we must consider that we took 3 kinds of lipids, and aside from them, there are also triglycerides and other kinds of lipids present in the brain. So the yield was reasonable but not perfect. So as a recommendation, in the future experiments, these factors must be taken into account. So that's the end of my report. Thank you, everyone. And comment down below if you have questions, and I will answer it for you. Thank you.